In this series of videos, we're going to be looking at the actual mechanisms that lead to evolutionary change across multiple generations. Now, in order to talk about what these mechanisms are and how they work, we need to understand how to detect whether evolutionary change is occurring, which requires us to understand a concept known as genetic drift. Now, genetic drift is defined as simply any observable change that occurs in the allele frequency of a population. Now, if this all sounds needlessly verbose, the easiest way to demonstrate genetic drift is by actually looking at how evolutionary biologists study evolutionary change across multiple generations. So we're going to be doing this with the butterfly population that we see here. We can clearly see that there are two alleles, two different forms of the gene that codes for wing color. This we will call the blue allele, and this is what we will call the red allele, just for the sake of simplicity. Now, if you are an evolutionary biologist, is it realistic for me to tell you, go out into the wild and look at every single butterfly in this population and tell me what percentage of them are blue and what percentage of them are red? If I told you to do that, the only smart answer that you should give to me is absolutely not. I am not wasting my time going out in the environment and chasing after butterflies. This is an obviously stupid waste of time because butterflies have the tendency to not sit still, and butterflies have the tendency to hide from humans if they know what is good for their very survival. Therefore, what evolutionary biologists do instead is they collect a sample of the population, which is going to represent approximately what we think the allele frequencies are. So in this case, the sample size of butterflies that we've collected is 8. We can see that 5 out of 8 of these have the blue allele, and 3 out of 8 of these have the red allele. Now, is this sample automatically representative of the allele frequencies in the overall population? Obviously not. So chances are, rather than having you being the only evolutionary biologist that is collecting this sample, chances are there would be other biologists that would go out and collect the same sample size. So they would be collecting 8 butterflies, and they would see what the allele frequency for their samples are. Now, what we do is we collect these and we make identification marks. So this would be butterfly number 1, this would be butterfly number 2, this would be butterfly number 3, etc., etc. And then we would release these into the wild and allow them to survive and reproduce. And after one generation, so after a year, because butterflies reproduce once every year, we would see whether the allele frequencies are the same or whether they have changed by simply collecting a second sample from the next generation. So we would be taking another sample of 8 butterflies, but now all of a sudden we see that the allele frequencies are different. So we can see, if we look at the allele frequency of our offspring population, we can see that all 8 of them are of the blue allele and 0 of them are of the red allele, which means if this is representative of a change in the population, this means that something has happened that is preventing butterflies with the red allele from surviving or from being able to reproduce, so that butterflies with the blue allele are now the more common variety of the 2 different alleles, suggesting that something is allowing them to survive better. Now, what exactly is causing this difference? Well, we don't know. There are actually a number of patterns that genetic drift can help us predict. So genetic drift, before we move on to those, is simply the observation that allele frequencies, how commonly each phenotype can be observed, changes across multiple generations, as we see with the difference in red butterflies and blue butterflies. There we go for a tongue twister. So one possible explanation for this actually has nothing to do with evolution. It's simply what we call sampling error. Now, in this case, let's take a look at this beetle population. This beetle population has two different alleles, green and golden. And let's say that you are not the only evolutionary biologist who is going on a bug hunt to capture beetles. Let's say there were 4 of you. Now, is it likely that all of you are going to get a sample of beetles that will have identical phenotypes in each sample? Well, that would obviously be no. So sampling error is simply the difference in allele frequency based on which particular sample was collected, and this difference simply exists based on which organisms were actually captured for study during the process of sampling, and this is one possible way to explain why there is a difference in allele frequency even across multiple generations. What does this tell us about evolution? Well, nothing on its own, because the difference in samples is simply due to random chance of which organisms in the population were captured by humans. However, there are actually 2 explanations behind differences accountable by genetic drift that can occur as a result of natural selection. So let's take a look at this population of butterflies that have clearly 3 different identifiable phenotypes, white, orange, and brown. Now let's say that these 3 butterflies decide we don't want to live among the ancestral population anymore. We are going to migrate to a different area because we don't want to have to compete with all of the other members of our population for food. So we are going to migrate to a different location and therefore become a colonizer group. Therefore, this small group here becomes an entirely separate population because now the genes that they have, the DNA that this migrant population has, can no longer interact with the ancestral population because they are separated geographically. But we observe that none of the white butterflies were part of this migrant population, and therefore it's unlikely that any of these butterflies in the colonizer group would produce the white phenotype because that phenotype wasn't represented in the original migrant population, and therefore the gene pool, the availability of different alleles, is going to be much smaller than the original population was, simply because only specific organisms migrated away from the original population. And this is what we call the founder effect. And if the founder effect has actually happened, we would detect this as genetic drift because the allele frequency of the colonizer population would look very different than the ancestral population. Now, kind of the opposite of the founder effect is what we call a genetic bottleneck, also simply called the bottleneck effect in genetics. And this is really the opposite of the founder effect because this change doesn't affect a colonizer population, but affects the original population. So in this case, we're looking at frogs. There are 3 phenotypes, red, yellow, and green, and let's say due to some unforeseen tragedy, the red population dies, possibly due to some sort of natural disaster that favors, if I can spell the word disaster properly, that favors the selection of one specific allele over another. Now, in order to explain what happens here, can the red population reproduce if they are dead? Obviously, that answer is no, and therefore the genes of the red population have now been removed from the gene pool, and therefore it is much, much less likely that red frogs will be represented in the descendant population several generations down the line, because this natural disaster saw to their removal from the gene pool, therefore creating what we call a genetic bottleneck where only certain alleles are represented in a descendant population. Now, is it possible for us to identify each of these phenomena just by sampling and just by identifying genetic drift? That answer is obviously no. So genetic drift can simply be used to detect or identify whether allele frequencies are changing or whether allele frequencies are constant, but cannot identify directly what the cause of the allele frequency change is. That would require study of the environment and of the population as a whole. So if we wanted to identify whether the red population was removed due to a population of blue butterflies migrating away, or due to some sort of selection pressure killing just the red population so that we get a bottleneck effect, we would require further study of the population to identify what type of selection pressure was causing this change to occur. So genetic drift is simply the observation in a difference in allele frequencies in the population across multiple generations, but the actual cause of this will be the subject of study for the next video in this series. So join us for the next video where we talk about the different mechanisms that can lead to evolutionary change over multiple generations.